So, welcome back to the 12th class of this series. So, we are into the third week. As I mentioned in my earlier class, that from this section, we will be talking about the pathogen as well as the sensor simultaneously. Of course, we will discuss some of the in-depth detail of the design of the sensors later, but at this stage, definitely we will go through all the fundamentals. So, in the last class, we concluded about the basics about the causative agent of anthrax and how it spreads, how it comes inside the human body by cutaneous or surface contact, inhalation, or by eating something which is contaminated with the microbe. Apart from it, we talked about how the people who take drugs could get contaminated with anthrax, but this is something which is predominantly observed in Europe, but still has not been seen in United States. And then we talked about a newly emerging anthrax situation happening to people who are working in the welding industry, steel industry and iron industry. And then of course, we talk about the 2001 anthrax attack in United States and how that was orchestrated and how that whole case concluded in the year 2010. So, from here today we will talk about the basic sensor modules which are used for anthrax research. So, to start off with, if we look at how anthrax looks like. So, this is the vegetative cell as I mentioned, and you have the spores. And I told you the biggest challenge is how to identify or sense the spores, because the vegetative cells are growing. So, when they are growing, they are secreting a lot of metabolites and there are a lot of active metabolites on their surface. There are structural aspects, there are functional molecules. So, it is far more easier versus a spore which is dormant. At this stage, it is purely dormant and there are hardly, so it is almost like a silently sitting there. So, most of the expression or the surface features are not there at that stage. So, if you have to detect them, you have to pick up signals which are extremely unique. And the other challenge is that when you are trying to detect spores, you do not want to crush this, because if you do so, the possibility of spreading the contamination increases. So, what are the target elements we are looking at? One of the target elements look at is the surface proteins, a signature for surface proteins, DPA, okay, DNA, and the toxins associated with it, or any other features which we are yet to discover, or they are in minor concentration, but not been tried out. So, what we need then essentially, we need a extremely sensitive sensor which could detect nanoscopic level of toxins, DNA, surface proteins, DPA and others. So, most of the biosensor world works with some of the basic principle of sensitivity at extreme low concentration, second thing is very precise signal with signal to noise ratio should be extremely low. So, almost a noise free system where you can pick up the signal, and the signal should be unique unambiguous, or at least we should have an idea what are the ambiguous element in such signals. So, what are the different kind of sensors which are being used or are in the process of development? One of them is lateral flow immunoassay. When we talk about the immunoassays primarily involve antibodies. Antibodies which are developed against Bacillus anthracis. Any form of immunoassay, any form of immunological assay will target the antibody molecules. Then you have a DNA or protein chips. So, if I go back to one of the very earlier slides, remember, that will help you to understand. Okay, here it is. So, if you look at this structure, you have the DNA, RNA structures, and then you have these plasmids sitting there. Then you have these different kind of proteins which are there. Okay, you have the lethal factor, you have extracellular polydeglutamate, the PGA factor. So, these are the target molecules. So, coming back in the present slide, which is kind of a follow up of the other slide, is this. Either you have a DNA protein chip, then you have the DNA sequencing, the DNA which are present in them if you could sequence. So, it will be a kind of a pattern matching. Okay. You match the DNA sequence. Then you have mass spec, Raman spectrometry and spectroscopy techniques. These are mostly used for toxins and other molecules. Then you have optical sensors, which uses some form of either fluorescent probes or some other light dependent mode, which gives a unique signal. Then you have the electrochemical sensors, where any of these target thing, the surface protein, DNA, toxins and others, or DPA, all these things gives a electrochemical signature. So, when we talk about electrochemical signature, something like either these molecules donate an electron to a receiver or they accept an electron from a receiver, and by virtue of that electron transfer, there is a quantification of this transfer of electron from one chemical to another. So, in a way, what you are trying to do, you are extracting the electrical signature from a chemical reaction. It could be vice versa. By giving an electrical impulse, you change its chemical nature and you record that particular chemical signature, or vice versa. In electrochemistry, it is basically you are deriving the electrical signature during a chemical reaction. So, say for example, say one of these toxins, so it say donates an electron, you capture that, or vice versa, that it accepts an electron and it gets reduced, you capture that signature. But that has to be a very unique signature, at what potential or at what voltage this whole phenomena is going to happen. Okay. So, next one is the nanosensors. Nanosensors is not something out of the world. These are basically all these different kind of sensors when they are using some form of nanoengineering tools or using some form of a nanomaterials during its manufacturing. So, let us summarize. You have lateral flow immunoassays, DNA protein chips, DNA sequencing, mass spec and Raman spectroscopy, you have optical sensors, you have electrochemical sensors, you have the nanosensors out there. So, now let us a little bit more into that. So, when we talk about the targets, we have the vegetative cells, we have the spores, we have the DNA, we have the toxins and the surface proteins and DPA, etc. So, the recognition elements of these are, as I talked about, monoclonal antibodies, then these are the fragments of the antibodies, then you have the peptides, you have the aptamers, and you have the phage display. And we will talk individually about them, so do not worry. Here I am just giving you the scheme of how the biosensor configuration will look like. So, you are trying to identify these elements. So, if you could recognize them, because these elements gives a, as I mentioned, unique signature. So, you have to know which are the elements which will give you a very, very unique signature. So, these are those molecules which will give you a unique signature. Now, in order to study these unique signatures, you need methodologies. What are the methodologies? QCM, quartz crystal microbalance. This will be the first one we will be talking about very soon. SPR, surface plasmon resonance. LFIA, we talked about lateral flow immunoassays. Okay. Polymerized chain reaction on a chip, PCR on a chip. Then you are using different kind of nanomaterials which either conjugate with these different recognition elements and gives you some kind of a signature which is unique to this situation. So, different kind of nanomaterials which are being used. Okay. Now, what kind of signal output are we looking at? So, there is a signal generation device, there is a recognition element. Signal output could be a fluorescent output, it could be a change in color, it could be a DNA amplification, or it could be a electrochemical, SPR angle, piezoelectric, Raman, EIMS, etc. So, these are the signature output. So, if you look at the whole field of designing of biosensors for biosecurity, and you realize that you have to have a comprehensive understanding of biology, especially the molecular events which leads to generation of these different recognition elements. Then after the biology and the biochemistry of it, and of course, the microbiology of it, you have to understand the basic physics of different devices, because these are all physical devices when we talk about quartz crystal microbalance, surface plasmon resonance, even partly the immunoassays which are being done on lateral flow, PCR, these are physical processes. So, you have to understand the physical devices which are involved with. Then you should have a very thorough understanding about how you process these signals. Signals will come, but you should have an understanding between what is noise and what is signal. So, you should be able to detect the background noise and pick up the signal which is relevant in your case study. Otherwise, you will have tools at your disposal, but you will not be able to really achieve much out of it unless you understand how really to interpret data or extract information from these different tools. Okay? So, from here I will move on to this one. So, we have already talked about what are being produced. Phage display I will come back later. So, I will start with quartz crystal microbalance. Thank you.